That. Okay, Megan, let me know when YouTube is up and running. Thank you, everyone. Hello in the room. Hello on YouTube. We'll get started in just a moment. Refresh that. And YouTube is good. All right, welcome everyone back to the next Water, Wetlands, and Watershed seminar. Today we have Dr. Julie Meyer. So Julie is an associate professor in the Department of Soil, Water, and Ecosystem Sciences here at the University of Florida. Dr. Meyer is a marine microbial ecologist, and if you don't know what that means, hopefully you know by the end of today. Her research focuses on how microbial communities influence the health and resilience of marine ecosystems, mostly corals, but also other stuff, sponges and other seagrasses, okay? And her lab examines complex relationships between microbes and their hosts. She earned her PhD in microbiology from the University of Illinois and did a postdoc at Hawaii and also at UF.
That's not me.
That's not you? Okay.
I can.
Did you ever go to Hawaii?
No.
No, all right. So Dr. Julie Meyer is planning to go to Hawaii on vacation in about a year. But here she began exploring the role of beneficial microbes in coral reef health. That part's right? Yes, okay. And since joining UF has helped reveal how microbiomes influence threatened marine habitats and hopefully strengthen ecosystem resilience in the face of climate change. So Julie, the floor is yours.
Thank you for the introduction and the invitation. I'm really excited to tell you today about one of my favorite projects, if we're allowed to have favorites. And so first thing I want to do is start with my acknowledgements, because the scale of this project would be impossible without multiple disciplines and multiple institutions collaborating together. And so this whole idea of probiotics for corals originated at the Smithsonian Marine Station with Dr. Val Paul and Dr. Mike Ushijima. And they are instrumental in, isolating the bacteria, doing all these testing for antimicrobial activity. With the help of Dr. Brian Walker's lab at the Nova Southeastern University, did a lot of many hours of field work to apply probiotics, to monitor, and things like that. And so my lab's role in this project is to sequence the genomes of those probiotic bacteria. So we really want to characterize what we're going to be putting on the reefs, and also to characterize what changes happen to the microbiomes. And I've had the help of a series of technicians, including the four shown here. And I wanted to add that Jessica Tittle and Monica Schroll both became, after they were technicians for me, became PhD students, and they just finished their degrees in August. So thank you for all of that help. So to talk about this research, I first wanted to set the scene for how this came about. And so I want you to kind of think about, what problem would you solve if you had the right microbial tools? And I like to tell my students, and probably Aaron may have remembered, that we are in a golden age for microbial ecology. So after a few decades of sequencing stuff and doing all these surveys in the environment to see who's there, what kind of genes they have, we're at the point where we can actually start testing ideas and using those microbial tools to change the environment, improve agriculture and things like that. And so I'm showing you here is a beautiful, healthy reef. This is in Little Cayman. Unfortunately, the reefs in Florida don't look like this. And to give you a sense of scale, these sea fans are probably like 2-3 feet across. They're pretty big. So this is a large reef. And for these stony corals that our ecosystem engineers are reef building, they depend on their associations with microbes. So both their photosynthetic eukaryotic symbionts, their microalgae, and their bacteria. And both of those components help provide nutrients for the coral. And this is a common theme. So whether these bacteria are in the soil next to the roots of plants, or whether they're in our guts or associated with the surface of corals, what they're doing is helping to make nutrients bioavailable to their hosts. And this is especially important in oligotrophic tropical coral oceans. So microbes are there to help us process our food. The other thing that they can do especially bacteria, is that they can make antimicrobial products. And so what they can do is protect us from pathogens. And again, this is a consistent theme, and we can apply this in lots of different ecosystems. But before I talk about coral probiotics, I can't not talk about the biggest threat to coral reefs, which is warming oceans. And you'll see that the top 3 lines here are the highest temperatures that have been recorded in the global surface ocean, are all in the last 3 years. And probiotics is not going to fix this. We have to address carbon emissions. Otherwise, they're all going to be gone anyway. And the reason for that is because corals have a very limited temperature range that they can tolerate, these tropical stony corals. And in addition to being, stressing them out, which leads to bleaching and mortality, for the ones that do survive, they're also much more vulnerable to things like coral disease. And so this is just a large scale disruption to them that can lead to lots of bad consequences. And collectively, coral diseases tend to be exacerbated by heat waves and things like that. One of the most problematic is this stony coral tissue loss disease, and I could probably talk a whole hour just on the disease, but I'm trying to refrain from that. It's was first observed in Florida in 2014, so we're now 11 years since it started, and since then it has also moved around the Caribbean. And what I want you to know about this disease is that it's predicted to be not only the most lethal coral disease, but the most lethal ecological disturbance that has ever been documented in the Caribbean. And so we are losing substantial coral reef tissue and area because of this disease. So, like I said, it's been 11 years. I want you to know that I said, what have we done about it? And at this point in Florida, we have finished what we could do. It basically has burned through our reefs. But before it did that, we did several things. We recognized the magnitude of the problem. And so we made a very concerted effort with the help of lots of people to do a rescue of corals before the disease got to them. So these stony corals are boulder type corals. Multiple genotypes from multiple species, all the vulnerable species were collected by divers and shipped off to different aquaria across the US. Mostly in the Southeast, but those aquariums are still maintaining those corals without any remuneration. So this is a very generous on their part. But we have that genetic bank to find new solutions. There has been substantial amount of research characterizing. The biggest idea is that what might the pathogen be? We still don't know. I think we're getting close, but again, that's a talk for another day. But what I want to focus today on is the intervention side. So because of the scale of this disease, in Florida in particular, where it started, the state has not only allowed access to protected species in protected marine areas, they have also allowed us, they have actually funded the intervention project. So on the left, you see a diver applying amoxicillin paste. We know it is very effective, but it is temporary, right? That amoxicillin paste doesn't only last a couple days. Then the antibiotic dissipates, it's broken down in the water column. And so those colonies can get repeatedly diseased, and so you have to go back and apply it. In addition to our concerns about using amoxicillin on a reef, because it's important to human health, we don't want to keep, we don't want to encourage resistance in the environment. We also decided to go with an alternative method, which is to use probiotics. This is what you see on the right. And the idea is that we will find bacteria that will make antibiotics for us that aren't involved in human health. And so how did we even get to this spot where we thought this was possible? Initially, we were trying to find the pathogen. So basically, we had transmission studies in the lab where transmitting disease, trying to see how fast it progresses, isolating bacteria from that to test as individual bacteria to see if they cause disease. And in this process, there was one coral colony in the lab that would not get sick. Now, this was unusual. Like most corals, if you expose them to CTLD, will die. And so this was an aha moment, right? Here's a coral that won't get sick. What is growing there? And so we isolated bacteria off that resistant coral, and it led to this large investigation with coral probiotics. Over several different years of searching and funding, the Smithsonian Marine Station ended up isolating over 4,000 different bacterial isolates that demonstrated that we could then screen for antimicrobial activity. Some of these were more effective than others. We sequenced the genomes of over 70 different isolates, which is this is a genetic resource that that is unusual to have, dozens of coral-associated bacterial genomes. And ultimately, we landed on one really exciting star strain, which is MCH17. I like to show the colors on the bacterial cultures on the right, because a lot of antimicrobial bacteria make pigments, and it's actually the pigments themselves that are antimicrobial. And so kind of an easy shortcut, if they make pigments, hey, let's try that. Plus they're pretty to look at. So our star MCH17 strain shows this antimicrobial activity, and an example of that assay that we use is on the left. So what you do is you take this agar plate, you spread on a lawn of a victim strain, and then you spot on your probiotic bacteria. So what you see is MCH17 in the yellow, 2 spots of that. And around MCH17, there's a clear zone where that bacteria could not grow near it because it's making something and pumping it out. And we were able to try this out in tanks with chunks of diseased coral. On the top, you can see if we leave a chunk of coral with SCTLD untreated, about a week later, it's dead. And there's some color left on the far right, but that's just rotting tissue. But on the bottom, you can see if we add the probiotic to the water, not only, that disease just stops. And we also found that in these transmission studies, that if you pre-treated a tank with a healthy coral fragment and then added a disease fragment, you could not transmit the disease. And in fact, that disease wouldn't start to heal. And so it can also prevent infection. Okay, so we went to 3 different kind of aspects of like, okay, we want to take this in the field. What do we need to do? So we looked at their genomes and also their chemical, I should say, for MCH17, we looked at its genome and its chemistry. We did some lab trials, and then we finally went to field trials. So what are we looking for in the genome? How do we find a good probiotic for disease? And remember, we're searching for something that's making an antimicrobial product. And when we talk about antimicrobial products, antibiotics, what we're really talking about is secondary metabolites or natural products. And so these are things that the bacteria can make that it doesn't need for its everyday metabolism. So these are extras, we call it accessories, right? They don't have to have them, but if they do, it might give them an advantage. And so this is where we have a beautiful supply of novel chemistry. So these are some examples of the kinds of natural products that these things can make. So what makes MCH17 so special? We were able to identify at least 4 different kinds of natural products that are antimicrobial, and sometimes multiple versions of it. So the coromycins, we know that Pseudoalteromonas MCH17 can probably make 3 different kinds of coromycins based on its genome. Pseudoalterins, there's 2 different sets of genes for pseudoalterins. And in the presence of with meridocin in the presence of the amino acid lysine can make hydrogen peroxide, which is what I've shown at the bottom there. And they also make tetrabromopyrrole, which a lot of Pseudoalteromonas strains can make. And we're interested in this not only for its potentially antimicrobial properties, but also because it's used as a chemical cue for coral larval settlement. So MCH17 can make a lot of stuff. And what I want you to see is that it can target a lot of different types of bacteria. So broad spectrum, some specifically for gram-positive bacteria, some for gram-negative. The other advantage of having the genome is not only saying, okay, well, here's what they could be doing, here's how they, oh, actually, one more thing I want to say before we move on. I have stars on poromycins and tetrabromophyrol because we were able to also isolate those chemicals from the cultures of MCH17. So not only does it have those genes, it also does make them in culture. And so we focused on poromycin because it gave us a way to have a unique biomarker for this strain. So remember, this is part of the accessory genome, so you don't have to have this. So we won't confuse it with other pseudo-ultramonas because there's plenty out there in the marine environment. And what you're looking at here is along the top is the poromycin biosynthetic gene cluster. And in particular, those green boxes are very large genes that assemble this poromycin product. And so this is what a lot of these natural products look like. They're big complex molecules, and they have a lot of genes that are used to build them. And so what we did is we developed a genetic target based on, I believe, the little gene called the number 23. That's our unique genetic tracker for that strain, right? So one of the reasons of sequencing that genome is so we'd be able to track when we put that strain back on the reef. So in addition to testing the efficacy of the probiotic when you add it to the water, we also wanted to know what happens if you just give it to healthy corals. And so what we did was we took 4 coral genotypes, split them up into 7 fragments, and the idea was that we take one fragment at different time points after adding the probiotic. So we just want to see, does this have any detrimental effects? And then we monitored it for about 4 weeks. And what we found is that, well, I first want you to know is that each of these columns represents the microbial community for one of those fragments. So we have 4 sets for the 4 different corals, and we have 6 different time points. And what you should see, like, don't worry about who's there or not there, is that they're all different, right? There's a lot of variability in the coral microbiome, and we expect this. So we're not surprised. What I was really looking for is, does that pseudo-ultramonas, that probiotic strain, does it bloom? And the answer is no. So again, this is just looking at the whole community. So if we survey the whole community, it's not there as a abundant member, but we can also use our genetic tracker to find it in our samples. So we looked at both the water in the tank and also the coral tissue. And so we can detect it sometimes, but at very low levels. So there's a couple exceptions. What you're looking at is copies of that poromycin gene per nanogram of DNA. And so what you're really looking at is just low, the biggest one was 20 copies per nanogram of DNA. Most of the stuff in the tissue is barely detectable. So it's there, but it's not establishing, it's not blooming. So under our controlled lab experiments, we know that MCH17 makes antimicrobial products. We know that those products stop disease progression. We know it doesn't bloom. And I didn't show this, but we also tested against healthy specimens of 6 species total. And so that adding that bacteria to the water didn't affect any of them adversely. So now we're ready to go try this in the field. So the field trials ran over 2.5 years, and it was really hard to know during that time if it was working. So it's a long process, but we recently published the results of this study. But it was a long wait to know, is it working, is it not working? So what we did is at the beginning, we sampled the microbiome before any probiotics were added. 2 treatments were added in the field. We sampled 2 weeks after those treatments ended, 3 months after the treatments, and then there was another probiotic treatment later. And in addition, there's photographic monitoring of the whole colonies for 2.5 years. So to be able to know what's happening with those colonies. There are 5 different treatments applied. And what I'm showing you are 2 examples. So the bag treatment What you're seeing on the left is the diver has this plastic bag, it's weighted down, it surrounds the whole colony, and there's a syringe with live bacteria that is gonna be pushed into that bag. Bag sits there a couple hours, the divers come back and remove it. So this is a systematic treatment. It's treating the whole colony at once. And we don't want to leave that bag on there for a lot of reasons, but we also tried this paste. And again, the live bacteria are mixed into this paste, it's sodium alginate. It breaks down after a couple hours, and we're applying the bacteria right to where the disease is happening. We also did some nice controls where we just applied the bag with no bacteria in it, applied a sterile paste. And then we also monitored corals and sampled corals that had no treatment at all. Okay, it's a little bit complicated, but what I want you to see is that these are PCAs. So we're looking at how similar microbial communities are to each other. So the closer they are, the more similar, the further away they are, the more different they are. And each point is a community from a coral sample. We sampled both healthy tissue, healthy colonies, and diseased tissue so we had paired samples from affected colonies. And the take home here, I want you to see is that treatment is the color. The treatment doesn't matter. The biggest effect on microbial community structure was time, like the sample date, and also health. So there's a difference between disease and healthy tissue, which we expected from all of the studies of the disease previously. But what I want you to see is that at 2 weeks after the treatment and 3 months after the treatment, there's no effect of the treatment on the variability within the microbial communities.
So each dot is like, basically like the composition of the microbes. And the dots that are near each other have similar composition.
Mhm.
And the colors of the treatments, and the black is no treatment.
Right.
And the colors are treatments.
Yeah.
And they kind of,
They're all
okay, all
big
right,
blobs.
Yeah,
Yeah.
okay.
I mean, I could put them all in one and then you'd really not see anything.
yeah,
So I separated by time because that was such a strong signal. And so this is testing the condition or the treatment, controlling for time, because time was definitely important. But all of these effects are only explaining part of the variation. So condition is only explaining about 5 to 7% of the variability in the microbiome. Date, the sample date was explaining maybe 15%. So by far, most of the variability is just background. It's like something we're not measuring in this treatment, in this study. And that's just background. It could be related to the genotypes of the colonies. It could be all kinds of things. So microbial communities are variable. And so sometimes it's hard to pick out patterns from that noise. If we pick out just the pseudo-ultramonas out of those surveys, so out of all those communities, we picked out all the pseudo-ultramonas, and this is based on 16S ribosomal RNA genes, so it's a taxonomic marker. We won't know if that's a probiotic strain or not, but what we saw is that 2 weeks after treatment, there was a bloom of pseudo-ultramonas relative to other times of the year, other sample times. But what you should see is that in that middle bar, the treatment has nothing to do with it. So it doesn't matter whether it was probiotic bags, control bags, the treatment didn't affect how much pseudo-ultramonas. Whatever was going on in the environment in October is what favored pseudo-ultramonas at that time. And still, it's not a huge part of the community. It's 10 to 15% of the total bacterial community. And you see in January that settled down. So this is based on our survey data. But again, we have that tracker, right? So if we use our poromycin tracker gene, we're getting very low copies in August and October. And we're really just getting a couple copies in our different DNA extractions. But in January, we had a lot more. And again, it's not tied to whether we treated it or not. For whatever reason, MCH17 just seems to be a little bit happier in January. So I think that's reassuring. So it's still not affected by the treatment itself. We're not changing things drastically. It's not blooming. And you say, well, it's higher after 3 months. But don't forget, we got this bacteria from this area, from colonies of coral in Broward County and we're doing these treatments in biochemical. So presumably MCH17 is part of the natural community. Now, don't forget, I said we also did this photographic monitoring for 2.5 years. And what this consisted of is having the camera set on like the, click, 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 and you swim slowly around the coral colony. And so you take a picture of all the sides of the coral, and then Kelly spent many, many, many hours putting together these 3D models to calculate what is the total tissue area for those colonies. And so we had a lot of data. This is why it took so long to figure out, is it working or not, right? To compile that and do statistical analysis to see, does the treatment help protect that tissue? Because ultimately that's what we're trying to do. Long story short, the bags seem to work better than nothing. So if we look at on the left, we have the proportion of total tissue area. So they all start at 100%. And then some of them go down to about 35% tissue left at the end, for especially like those, the black line, which is here it's background control, obviously the no treatment, the things that were just left to die while we watched. So basically at the end of the treatment period, the monitoring period, we had the highest tissue left for colonies that were treated with the bag.
You just remind us what's PP, CB, CB.
Yes, so that dark orange is the probiotic bag. Interestingly, the probiotic paste is the light orange.
Yeah.
And it did almost worse than leaving it alone. But the control paste and the control bag are somewhere in the middle. So doing something seemed to change the colony's trajectory. But that paste with the probiotic bacteria, we have some ideas that maybe it has to do with maybe the paste is too anoxic and Pseudo-Ultramonas doesn't like it and dies. And maybe it just kind of like triggers bad things when you apply the paste. So we didn't really test for like how long does the strain survive in the paste, because it was mixed right before you did in the field. Because you see the paste itself is not the problem. It's the combination of paste and
BC's background control with no
treatment. Yeah. So bag seems to work. It's very time consuming, and those bags are heavy because that weighted wire is hard to carry. Like if you've ever done diving, anything that's got surface area just has lots of drag. So you can imagine weighted bags treating multiple colonies. This is a lot of work. But the bags did seem to work. So at the end, our probiotic treatments, the probiotic Pseudo-Ultramonas can stop coral disease. The bag samples, some of them only lost like 20% of their coral tissue area instead of 75–80%. So we were able to slow coral tissue loss, and the effects were long lasting. So I'm going to just go back to this chart. The black arrows on the left are when the probiotic treatments were applied. And so that was day 136, and 2 years after that, it still stayed on that trajectory. So effects were long lasting, and the probiotic didn't bloom. It didn't alter the microbiome. There's lots of variability going on anyway. This probiotic treatment didn't seem to make a dent. We think we know what it's doing. It's making antimicrobial products, and probably that poromycin is probably a big player in that. But why does this work? I mean, you might be asking like, okay, well, the probiotic's like barely there. How is it working? I would suggest that it's disrupting the disruptors. So when this disease happens, a lot of marine diseases, not just coral diseases, are polymicrobial. So it's not one pathogen, one disease. There are multiple pathogens or pathogenic opportunistic kind of bacteria that come in, feed on tissue that's damaged. And so I think that this Pseudo-Ultramonas is just tipping the balance a little bit so that the native bacteria can take over, become more stable. The coral itself can make antimicrobial products. And so it might just be enough to tip that balance back to, let me have a healthy microbiome. So that's my suggestion. Okay, so we've learned a lot from what we try to do with these coral probiotics. And I think there are some take-home considerations that I would advocate for if you wanted to use probiotics in a different environment, a different system. And for that, I would stress that you definitely want to have native bacteria. So we could barely make a difference adding MCH17. If you try to use a probiotic that you got from a different environment, a different coral species, I doubt it would take at all, right? So if you think about humans, we take these you can buy probiotics at the store and take them every day, but if you stop taking them, it will stop being there. So you have to like constantly reintroduce it because it won't get established. So you're gonna want to try to get something native. It means a lot of work. You're gonna have to do that for every organism that you want a probiotic for. Transient is good. You don't want something to create a new problem, which we sometimes have seen with different like bioremediation things. You don't want to introduce something that's going to take over and become a new problem. You want to be able to show that it actually helped your host for whatever way that looks. And I suggest understanding the mechanism. So the more you can learn about their genomes, their genetic potential, what kind of products they actually make, the better. So you know why it's working. One thing I didn't talk about in the genomes evaluation is we also looked for virulence genes to make sure it wasn't going to, again, cause problems that weren't already there. So I may have gone, no, I have no idea what time it is. I'm going to finish on my last slide to again acknowledge my partners in this and my funding. And I definitely would like to hear from those of you in the room what you would also consider if you wanted to do probiotics in a new environment.
Right. Okay, so you did great. We have plenty of time for questions. Yeah. And we'll start in the room. Questions for Dr. Meyer? Yeah, Aaron.
So I think I understand the bag control treatment. For the paste control treatment though, what was that? Like what was in the paste?
So the paste itself is a sodium alginate. So it's kind of like just like a gel. And so some of the probiotic paste had the live bacteria added to it, and the control paste just had nothing.
Okay.
So it dissolves pretty quickly. And that was actually, there's a lot more to the development side of delivering a probiotic, right? Especially underwater. You want something that sticks for a little while, enough for the bacteria to transfer, but you don't want it to stay there forever. So both the amoxicillin paste and the probiotic pastes are made to go away. Yeah.
I don't know much about corals, but I was wondering if you ever considered kind of like the diversity in what was working where and what wasn't, having to do with grazers or other, macrofauna that may be moving things around.
Yeah, actually, that's a great question. So I was on one of the dives where they were applying the amoxicillin paste, and I could see fish coming along and chewing at that edge, like on the paste. Like, what is
Tasty.
It can't be good. I mean, but there are definitely documented cases of where grazing fish will transfer disease from one colony to another.
You might have said this already, but just so stony tissue coral loss disease, you said has burned through the reef. So can you just give us sort of like the status and trends report on Florida reef systems and
Yeah, so there's still boulder coral. So what the stony coral tissue loss disease impacts the boulder species. And so there are more than 20 species that are susceptible to this. It doesn't affect the branching, the properas that they're using in restoration. Unfortunately, the heat is what's damaging. They're more susceptible to the heat. So we've lost a lot of live tissue, but there are, in addition to the proper restoration, there are lots and lots of efforts now ongoing to get boulder corals restored. And so what they're doing, you can imagine if the tissue on these live, large, the ones that were once live, the skeleton stays there. And so you can, in fact, reskin them. So you can take some of that genetic bank, you can microfragment off of live tissue, especially resilient ones that are still left on the reef. The ones that are there presumably are fighting. And so you can, you know, cut off a piece of living tissue, grow it on these little pucks, and then just glue down these live pieces, and then yeah, the idea is you reskin that big large.
Do we think that the
Right.
time scale of regrowth versus like, there's like erosive processes, like
Yeah,
and do you need to then treat, like pre-treat? I guess if they're already sort of there, they're telling us that have the capacity for
yeah.
resisting, right?
I mean, for the fragmentation and the propagation of the resilient corals, they still are bringing them in to the lab to do that, and
Yeah.
they might lose whatever protective property it is. We're not at the point where people will quite let us apply probiotics in the nursery setting. I think that is a good idea, but again, it's kind of all that background prep. Each coral species probably needs a unique probiotic. But there are ways around it.
So you mentioned that you think the mechanism driving some of the behavior that you observed is disrupting the disruptor. Given how diverse I guess the microbiome is here, do you think it's organisms working in tandem to disrupt the disruptor, or maybe several disruptors working in tandem?
So the thing we're continuing to work on is figuring out what causes this. We know that from studies around Florida, around the Caribbean, that there are consistent groups that are selected in this disease process, and a lot of them are anaerobes. And part of that is because like once the damage starts and things start to rot, those guys will come in and just chew on what's left, right? And so we find that those are enriched. We still don't know what that first initial damage is. Like, is it just stress? Is it because it can be stress? I mean, where it popped up in off of the coast of Miami, it was after dredging, and dredging is a stressful event to corals because that sediment gets deposited on them and it kind of shades them, and it's a stress factor, right? And so is it something like that, stress them out, that coral can't keep its normal microbiome, and then these opportunists just come in. But importantly, what we know is that it's that similar groups are recruited to the disease. It's not like the one single kind, not one single strain. So it's kind of frustrating. Actually, that kind of hurt me again.
Yeah.
Well, my only question is, like, just how local is the strain? Well, obviously the strain is a result of a little bit of extra testing, but the original microbe, just how local is it to the Broward coral?
That's a good question. So Pseudothermonas as a genus are really abundant. We have ongoing work to do a comparative genomic study of Pseudothermonas, and that particular strain seems to be a unique species compared to what's been sequenced before. But I don't know if it has to be that strain, right? Because I didn't show that to you, but we have 30 something other Pseudothermonas in the lab, and they also make lots of stuff. Some of the other potential things that we screened for were just maybe they were good at attacking bacteria, but then you tried it on an Orbicella fabulata instead of a Montassia cavernosa, and it made that coral sick. So sometimes it's just about context.
So I've never heard of probiotics being used to treat corals. Is there anyone else that you know of in the Caribbean doing similar research to you, or as far as you know, is it just you and your team?
So that's a good question. So the idea for coral probiotics actually started in the Red Sea. And so they were using it for heat stress, like because the Red Sea gets really hot, those corals are adapted to it. And so they started trying to find, strains that were maybe help with that heat resistance. And that's Raquel Peixoto. She's from Brazil. And actually when she was in Brazil, she looked for bacteria that could break down oil, like from oil spills. And so basically I always say that she was the first one to say, hey, let's use probiotics for coral. In addition to her Red Sea work, In Florida, it's just that, yeah, Blake is the one that's spearheading other places in the Caribbean, so. Yeah, so very few places, and I would say the third place in the world that this is being examined is in Australia, but those groups are using more kind of a black box approach, so they're taking slurry tissue from healthy corals or resistant corals and using the whole thing, like they're not doing the isolation part. Yes,
Okay, like in a blender,
yeah,
yeah, okay.
So is death due to one microbe or is there a succession of microbes?
I think it's probably a successional thing. I mean, one thing we have considered is that what if it's a 1-2 punch with a virus and a bacteria? Like when we get pneumonia, it's usually a cold virus that starts something or a flu
Yeah.
that reduces the health of the host and then bacteria colonize, right? So it could be something like that. People have searched for viruses a little harder to screen for viruses. We just, we're scratching the surface of what we know about viral pathogens.
All right, well, YouTube is pretty quiet, although I know you do have to log in if you want to ask a question on the YouTube chat. But you often have a little hesitancy there. Any last questions for Dr. Meyer? All right, one last round of applause. Thank you, Julie, so much for joining us today. So next week, our speaker is Mark Masters. Mark Masters works with the Ag Water Policy Center up in Georgia and telling some interesting stories about the very long and protracted battle over water between states and how actually consensus finding is possible once you get landowners in the room and try to get sort of the politics out of it. So really nice story. And thank you again, Julie. See you all next week.